So there are several different cases for HTT in parasitic plants and their host. And a couple of years ago, they published a paper on Rafflesia and also some other cases on Cuscuta and Predigo. And right now, we have more and more evidence that the parasitic plant, since they are very close relationship with their host, there's a direct contact with their host. So there should be a lot more cases for the DNA transfer. So we can see some of the evidence here. So again, the evidence is from the comparison of the phylogeny. And this is the NAD1BC exons in Rafflesia. And on the left, that's the species tree based on MED-R, phytocon C, 18S ribosomal RNA. So it's Rafflesia is a parasitic, very famous parasitic plant. It's very big. It's parasite on vitis, a big vine in, well, it's in the grape family. So, but if you use the sequence of NAD1BC and you show that it's grouped with this yellow thing here, the yellow thing including tetrastigma is from VTC, that's the grape family. So that's its host. So this means that this particular gene piece or piece of sequences are derived from the host and then transferred to itself, the genome of the mitochondria. So this is a clear case. And also there are several other groups, including Daniel Nickren's group, study the several other parasitic plants, including these two, Apodanthus and Pilostylus. And those two has been put into the same family before in the Rafflesia. Anyway. So here we will see this is the phylogeny based on MED-R, mitochondrial MED-R. And these two, based on the species tree or other genes, they are grouped with these other taxa in both phases, which is Rafflesia. And in a MED-R phylogeny, it's quite distantly related. It's closer to the cucumber family, Cucurbitaceae. And Mitrastema, another former member of this parasitic plant family, is Mitrastema. It's closer to Ericaceae, but it's possible, since it's probably not belong to here, you have to compare it with other genes. And Mitrastema is a very unique plant. Well, you can find it in Taiwan, actually. And this plant, if you use the MED-R sequence for Mitrastema show the relationship of Mitrastema with Ericaceae. And ATP1 shows this species to be closer to Caryophyllates, which is another group of plant. So which one is right? Well, it's hard to speculate if you only compare those two. But if you compare, if you're adding like 18S ribosomal RNA, other cases, you'll find it. And here are the 6 different kind of genes. If you use the Mitrastema as an example, you will see the mitochondria MED-R. Okay, well, these are the same. Mitochondria MED-R group with Ericaceae, ATP1 group with Caryophyllates. But the small subunit ribosomal RNA group with Ericaceae in this case. So it's more likely that Mitrastema ATP1 here has a horizontal gene transfer event for the ATP1, but not MED-R. And for the other cases like these two taxa, and those two genes from mitochondria both show the horizontal gene transfer. So what is the significance of this? The idea is that even within the mitochondria, it's not necessary to have a mitochondrial capture in these cases. So only some genes in mitochondria was replaced by by some other genes due to the horizontal gene transfer, but many other genes retain within it. So how does this happen? We have the idea actually why the ATP1 was replaced but not the others. And this happened in, the things in green shows that it's likely due to horizontal gene transfer. So only some of the texts are, these are all parasitic plants, right, in this table. And only some of them show very special pattern for horizontal gene transfer in here.